RIKEN, and she's going to talk to us about spider silk. And it's very interesting. Personally, I hate spiders, but I'm happy for the things that they do. I know that they're very useful creatures, and I'm also very interested in spider silk. So I'm happy to listen to the talk, but not to see the pictures of the spiders. And so Alia has been living in Japan for a little bit more than 5 years, and she studied biochemistry in graduate school. And so I think she'll give us an explanation of the work that she's doing with spider silk. So please, Alia, go ahead.
Okay, thank you. I will share the screen right now. So, okay. Right. I hope you can hear my voice, right? Okay, good afternoon, everybody. My name is Nur Alia Octaviani. You can simply call me Alia. I'm from Biomacromolecule Research Team, RIKEN Center for Sustainable Resource Science. So today, in this nice opportunity, I would like to give a short talk about unraveling how spiders spin their silk. Okay, when we listen the word silks, I mean, what is silks actually? Silks is a natural protein fiber. It can be produced by the silkworm, and which can produce this silk cocoon, or spider, which will produce this spider web or spider silk here. So and commonly people always think that silks related with the textile, and that is true. So this is like one of the example made by the silk cocoon. So this is a silk saris from India, and this is a silk cape, which is made by more than 1 million golden orb spider in Madagascar. So that's really beautiful. And a very long time ago, actually, a doctor in Greece and also Rome, they used spider web as the wound dressing. So there are many types of the spider. I will not explain one by one. I'm not the expert of many types of spider, but one of the very famous spider, I'm sure that many of you knows about this. It's called Trichonephila clefata or Chorogumo. So it's found in Japan, Korea, Taiwan, and China, and this is also this type of spider is commonly used in our laboratory for experiment. So this is a member of a golden orb web spider genus. So if you have a look, this spider web is not so white, but it's more golden. So it has a golden color and like a bit more yellow. And the female body is much bigger compared to male body. So female body is 17 to 25, where the male body is about 7 to 10 millimeter. So interestingly, there are actually several types of a spider silk, and spider can have like up to 6 to 7 spider gland, and then which will produce the silk for different purpose. Let's say, for example, this purple gland, it's called aggregate gland. It will produce this aqueous coating of capture spiral. So this actually very important to keep the insect stick into the spider web. So this insect then can be food for the spider. Or this green gland, or called the flagelloform glands, it will form the core fiber of this capsular spiral in the spider web. Or this minor ampullate gland shows in this color. So it's become the will form this auxiliary spiral, and this orange gland which will produce the drug line and structural silk. So it will function as the lifeline, or it also like can hang the spider from the wall also, from the ceiling, and then this like a cylindrical glands, this blue one, so it will be outer silk of the egg sac, and this green is aciniform gland is become the soft inner silk of the egg sac, and the piriform gland is which will produce the cement for join the attachment of the spider web. So and mostly studied is actually this what so-called the spider dragline silk, and for the rest of my talk, I would like to talk more about this spider dragline silk. So why spider silk is very interesting, and one of the reason is first is biocompatible. Biocompatible means that it's not harmful for the living tissue, or it's not toxic. So and then after that, it's also biodegradable, and importantly, it exhibit excellent mechanical property. So if we compare this to the Kevlar, for example, and then this is a dragline silk, and this is nylon and silkworm in the stress strain. So this will describe the strength and also the elasticity. So we can see that this spider silk exhibit as the toughest fiber of all, compared like to Kevlar, nylon, silkworm, and silkworm cocoon. So, and you can see here this in this table, actually this for example, this Araneus diadematus, which is the dragline silk, also showed very high toughness compared to the Bombyx mori, elastin, nylon, Kevlar, steel, and copper wool or carbon fiber. Okay, so nowadays people process the silk into new biomaterial. So it's not only like for textile. So normally this like a spider web, for example, right, or the fiber solubilize, and then we can produce like many type of silk, but into new biomaterial, like a hydrogel. This is like a jelly, and this is the film, sponge, or nanoscale fiber, and even also still use this for textiles as well. And for that, there are many potential or application which can be used not only for like industrial, like bulletproof clothing, astronaut clothing, but also many of them can be used for biomedical purpose. For example, artificial skin cells carrying scaffold, like a surgical suture, because it's biocompatible, doesn't make like create the allergy or toxic, and it's also good for like a drug delivery or even just for cosmetic. And so what is the spider dragline silk? So how we can look at this structure of the spider dragline silk? So if we have a look into the spider web more detail, it's like a bundle of the silk fibrils like this. Actually, it's quite a complex structure, and like shown as this figure. This is actually based on the simulation. And if we have looked the modular structure of spider silk protein, there are three main parts of the spider silk protein. So this is the end part, and this is the middle part, and this is the C part. So the middle part is actually called the repetitive domain. So it's like a repeating unit. It's like a molecule repeated for 50 to 100 times repeat. And it's very long and very flexible. And while the end side and the C side is small, well, rather small compared to the middle part, but together it will form a very big molecule of a protein. So why we need to make artificial spider silk? We know that a huge potential application for this spider silk because of very interesting properties. But we can always collect a spider web, right? But actually, one of the reason is that spider has a cannibalistic behavior. So they tend to to kill one another and eat them. So that's the reason why farming spider is very difficult. And if we collect the spider web, of course, it is possible, but it's very time consuming. It will take many years just to make a very good textile. So how to make the artificial spider silk protein? So one way, we can use a chemical synthesis. In our laboratory before, we use the chemical synthesis to produce the spider silk, but unfortunately it's still in the short length of the spider silk, so we cannot synthesize like the full length of the spider silk. So in many cases, people use the recombinant DNA technology. So how it works, actually, we take the silk gene from the spider and then we put into the vector, and then we like inject this to to many different hosts like bacteria, yeast, plant, or goat. So the silk can be produced in the goat milk also. So our recent study published last year used the photosynthetic bacteria, marine photosynthetic bacteria, to produce the spider silk. So the principle is the same again. So we use the silk gene from the spider, and then we put into the vector, and then we transform that or inject that to the Rhodophyllum sulfidophyllum, this purple bacteria. So this is, if you have a look, this is like a 10-liter culture, and it can produce this, well, this is like a powder, but it can produce like fiber, which is like a spider silk here. And this is the fiber shown in this picture. And interestingly, this is really environmentally friendly because this is like photosynthetic bacteria. So it utilize light and nitrogen and also carbon dioxide from the nature. So it's actually environmentally friendly and also economical. So when we compare these mechanical properties of the native and artificial spider silk, so we can see that here, if you have looked this native drug line silk, like from Archaeopteryx fasciata and also Araneus diadematus, we can see it has a very good toughness, but the synthetic one is still not as good as the native one. So people still want to make it as good as the native one. So how we can do that? I mean, so far the artificial still the mechanical properties from the artificial spider silk still much less than the native ones. So yeah, the question that how to make this strong artificial spider silk, like, so we can use like this Spider-Man, so it can like hang and then jump from one place to another place. So one of the key is to mimic natural spinning process. So we need to learn from the spider itself how the spider can produce the silk. So for that, we need to have the comprehensive understanding about natural spinning mechanism. So what happened actually inside the spider gland? So this is just illustration of the spider gland. what happens? So actually in the spider gland, it's a pH gradient gland. So but the pH is rather neutral, but when it close to the spinning duct, it's getting more and more acidic. And here is also a high salt concentration, like a sodium, chloride, magnesium, calcium. And then when it close to the spinning duct, well, like this iron is more dominant, and this is getting less in the spinning duct. So what happened? So first of all, I just want to show what happened for this small domain, like the C part and N part. So N part is actually just like in well, monomer, or like alone in this uh, spinning gland. But when it close to the spinning duct, actually this end part is actually interact and connect the spheroid. But what happened with the biggest part, which is the repetitive domain? That's the middle part, because repetitive domain actually the biggest like dominate the length of the protein chains. So our study shows that it's actually very random coil structure, but it has like a small population like this, which has this type of structure. And this type of structure actually like helps for alignment. So with the help of the shear forces and this change in biochemical environment, so this from this it will form this type of a silk fiber. So that's actually the secret of this how soluble spheroid can be transformed to silk fiber. And then how actually ions. So we know that ions actually enables presence in the spider gland. So how ions enables spider spin their silk. So actually in the spinning gland, which has a lot of the sodium, this ions, sodium, chloride, magnesium. So it's actually prevent the interaction. So it keeps this become more individual. So this this ion actually make it soluble, so it because it prevent the interaction. And then, well, oppositely, in this close to the spinning duct, there are more of this ion, and this ion actually make it more interaction. So it actually can trigger the silk formation and finally can form this structure. So it can form the spider silk with a strong intra and intermolecular interaction. So yeah, of course, there are a lot of more of the secret that we still need to study. But yeah, for this time, I just want to share about this. I'm very thankful for, and I would like to acknowledge biomacromolecule research team in the RIKEN Center for Sustainable Resource Science. In particular, I would like to say thanks to Professor Keiji Numata, which is professor in the biomacromolecule research team, RIKEN Center for Sustainable Resource Science, and Department Material Chemistry, and in the Graduate School of Engineering, Kyoto University. And I would like to thank also Dr. Fumiaki Hayashi, Dr. Akimasa Matsugami, and Dr. Toshio Nagashima, which is from the Advanced NMR Application and Platform team in the NMR Research and Collaboration Group, Dr. Ali Malay, Dr. Hamis Craig, and Dr. Jian Ming Cheng, and Ms. Mamigoto from the group in the Biomacromolecule Research Team, RIKEN Center for Sustainable Research Science in Japan, and Dr. Chon Ping Fung in the Department Material Chemistry, Graduate School of Engineering, Kyoto University, Japan. Thank you very much for your kind attention, and I'm very happy to answer your question. Thank you.
Okay, thank you very much. That was very interesting.